We are.
You're perfect. Great. So I'm representing the genetics working group, and in Frank's opening, he pointed out that this was a new group for the grant, the continuation grant. I do want to mention that Frank and I were collaborators on an organic transitions grant previous to this, where we did initiate some work on rootstock breeding. So my group has a history with rootstock breeding, but we sort of had put that program on the shelf for a couple of years and then reinitiated it with this particular project. So our group's main goal is to utilize the genetic diversity in Solanaceae and Cucurbitaceae to improve plant function. We do have a central hypothesis that rootstock vigor and resistance to pathogens and environmental stresses will improve scion health and productivity. We sort of organized ourselves around three main goals, or three main hypotheses, I guess I could say. And I'm going to touch on a couple of these as we go. The approach that I'm taking is to present work from different team members and try to tie them back into these goals and hypotheses. My view of the flow of this is a little bit simplified relative to Frank's. I'm sort of more of a Venn diagram guy. But what I want to point out is that members of the genetics working group overlap with the Solanaceae production group, the Cucurbitaceae production group, but then some of these larger issues, extension and outreach, economics, technology, and post-harvest, encompass everything that we do. So, as I mentioned, one of our goals is to look at resistance, whether it's major gene resistance, non-host resistance, or vigor as an effective mechanism to improve scion yield and quality. On the Solanaceae side, we are mostly focused on tomato, but there is some work going on with pepper and eggplant as well. There's quite a number of diseases that people have looked at, and I'll feature some of these. On the Cucurbitaceae side, watermelon is the main scion, although there has been some work with melon in general. And then people in the group are working on verticillium, Phytophthora crown rot, and powdery mildew. I'm going to start by talking about some of the work that Carol Miles's group is doing at Washington State. They have been screening wild cucurbit accessions from the National Plant Germplasm Center, and specifically looking at resistance to verticillium. And the symptoms of that are shown here, scion symptoms. So the general approach is to raise seedlings of these accessions, inoculate them, do a root clipping to promote infection, and then transplant into the field. This slide summarizes some of the results from that work, especially the master's student thesis work here. And so what we're looking at here is a susceptible control, and then area under the disease progression curves. And so then on this end, you see that there are a number of PIs, or plant introductions, with putative resistance. So they've identified 7 accessions from this species that are showing potential resistance, 1 here, and then 5 from the Moschata, and those are all going to be tested again in this year. And I'm apologizing because I realized I cut off the species name here. That was my fault, not Carol's. Okay, I'm going to now kind of segue into some work that Shakar's group is doing with the USDA in Charleston, South Carolina. So they're interested in screening the Moschata and the Maxima plant introduction collection for resistance to Phytophthora crown rot caused by Phytophthora capsicum. So you see here plants just after inoculation and then 2 weeks later, pretty devastating screen. The results are fairly clear here. But this is where the fun starts, right? So so what Chakra's group has found is that the Maxima accessions, for the most part, were susceptible with a few exceptions. There were 4 PIs that were rated less than 2, and so you can see those here. In contrast, you have the susceptible control here. But when we go to the Moschata accessions, there were 5 that rated less than 1. So again, just check the scale. The scale is the same on both graphs, so the Maxima and the Moschata. So this group has identified some potentially resistant accessions. Now, I just want to make sure everybody's on the same page here, is that these tend to be outcrossing, so the accessions have some residual genetic diversity. So one of the things that his group has been doing has been carrying those selections on in order to fix that resistance. And so there are several new sources of resistance to crown rot that have been discovered. That material is being inbred, and there is a goal of developing some resistant interspecific hybrids from this screening. Still sticking with the work that's been done by Chakra's group and the USDA group in South Carolina, I'm fascinated by this. So there are powdery mildew resistant bottle gourd rootstocks that provide resistance to watermelon scions. So this is one of the few instances that I find are convincing that a rootstock can provide some level of resistance to a foliar disease of a scion. Okay, and so some of these powdery mildew resistant bottle gourd rootstocks are now available to cooperators, and so there are both inbred rootstocks as well as hybrids. These bottle gourd rootstocks are also resistant to fusarium. Okay, moving on, I'm going to switch gears to the Solanaceae. I want to thank Jean Miao for this slide. Yeah, so soil-borne diseases of tomato, it's everything and the kitchen sink. Right, and so Jean provided me with a number of slides of soil-borne diseases that affect production in California. Some of these are problematic all over the country. So verticillium, there's a widely deployed verticillium resistance to what we call race 1, but race 2, or as I prefer to call it, race other, is showing up all over the country: California, the Midwest, East Coast, Florida. Fusarium, race 3 is proving to be problematic. There is genetic resistance to race 3, but that genetic resistance is associated with some negative characteristics related to vine and fruit. And so in some cases, it may be better to deploy this resistance through rootstocks. And so Aaron Roscoff's group at Forest Pierce in Florida has been looking at sources of fusarium resistance and basically demonstrating that the race 3 resistance is effectively transmitted through grafting. And so some of the issues of deployment of that major gene resistance go away when you graft. Fill this in. Her group has also been looking at this intersection between genetic resistance and environmental stresses. There's some other work that's going on looking at nitrogen use efficiency. That work's being done in North Carolina, but I just want to use this Aaron's work as an example of the sort of intersection between genetic resistance to disease and to abiotic stress. And so I'm going to fill this in. So her group has been doing some screening under different levels of salt, shown here. They're measuring photosynthesis. And at the end of the day, under high salt stress, we're still able to see resistance holding up in some of this grafted material. Okay. So verticillium, we have some putative resistance discovered in some of the Hawaii material and hybrids. We are going to continue to evaluate that material. Jean Miao and I are cooperating on a trial in California this year, and Frank has done some work in North Carolina. Bacterial wilt, there's a large number of team members that are interested in resistance to bacterial wilt. So this is a slide taken from a publication with Matthews Parrott as an author, Josh Freeman, and some of the other USDA folks. So here you see a non-grafted situation. It's pretty easy to score. And then grafted to a BHN rootstock with some resistance here. And then this is just a field epidemic. Dilipanthy and his group at North Carolina have been doing a multi-location test for resistance to bacterial wilt. And I'm just summarizing data from one of their locations here. So a key point is that there are several rootstocks, some commercial, some experimental, that are providing resistance. And the fruit size is not just high, but commercially acceptable for this material. Again, in North Carolina, there was a kind of a chance opportunity to screen for southern blight, and again, some of the Hawaii material and hybrids were showing resistance. Okay, I'm gonna move on really quickly here. So what I've just covered is screening of germplasm for specific resistances, and the take home is for both the Cucurbitaceae and the Solanaceae, novel resistance has been discovered with this project. But one of our goals is also to look at more generalized breeding themes. And in rootstocks, the commercial rootstocks are, at least for tomato, generally hybrid-wide crosses. And that has led to some issues with both uniform germination and also potentially with compatibility. The seed companies have really done an excellent job of dealing with the uniform germination issue by implementing some seed priming. What we wanted to do was kind of systematically look at this issue from two perspectives. So one is looking at hybrid seed production with both wide crosses and narrow crosses. And so I'm just showing a group of parents here. So there are cultivated types and then various wild species. And one of the questions that we wanted to ask is whether the genetic distance between parents was a predictor of hybrid vigor with respect to rootstock growth and scion growth. So we ran a number of trials. I'm just going to go through those pictures really quickly so you can see the way the trials evolved. And what we found is that genetic distance between parents is positively correlated with scion growth. Okay, so the more distant the parents, the more vigor was imparted to the scion. But at the same time, as I mentioned, that genetic distance is negatively correlated with things like seed size and germination. Okay, and so this is really what a breeder has to balance: improvements with negative effects. And so again, this is we evaluated close to 70 rootstocks through this system, and I'm only focusing on the ones sort of at the top. So you see we've got Beaufort and Maxifort here, so those would be our commercial checks. In this case, we were using a pretty decent scion, so the scion looks pretty good. Of the rootstocks that exceeded our checks, the ones in yellow all have some, what I would say with quotes around it, issues, either germination or grafting success, while there are several that also do well, and those are in the light green. Okay. So one of the things that we wanted to do is go back and see if we could identify selection points in rootstock breeding. And this slide basically just summarizes what we looked at. We looked at the success of crosses in terms of how much seed was produced, the quality of the seed, germination. We looked at grafting success and incompatibility, and then we took it all the way to field evaluation. And we're approaching this from the perspective of the heritability of those traits and the ability to make gain under selection. And so we think that we've identified, at least for tomato, selection points for rootstock improvement through breeding. Okay, so this slide covers the conclusions. And I just want to point out that so one of the benefits in working with a large team like this is there's the ability to leverage the personnel in this cooperative agricultural project. So germplasm exchange, expertise in disease screens, expertise in seed production and analysis are all distributed, right? So my role is to develop germplasm, and I'm distributing that to people for testing. Shakar's group is beginning to help Carol's group in terms of how to manage inbreeding and carrying resistant material forward. We found resistance to multiple soil-borne diseases of both Solanaceae and Q. cucurbitaceae. Some of that material is at the point now where there are actually inbred and hybrid rootstocks that are named. I just covered this, and so we can kind of go forward. One of the things that some people accuse me of being negative, but in our experience, grafting does not always improve yield performance in the open field. There are exceptions, and those exceptions are when soil-borne disease may limit production. When the scion is an heirloom, grafting almost always helps. And where there's season extension and we can carry the crop on longer, grafting almost always helps. Okay. And then this is my final slide, and I'm actually going to ask our advisory committee for some help. So coming up in April, there's going to be a meeting that's called the Tomato Breeders Roundtable, and we're planning on running a rootstock breeding workshop on April 5th. We're going to make this available as a webinar so people can take advantage of that, and I think Matt is going to distribute the URL for that. I'm looking for somebody who can speak about rootstock breeding from an industry perspective, and there's several members on this advisory team that might fill that role. I actually had somebody from VHN that was going to do that, but he backed out. He had somebody resign in his group, and he just feels like he's too busy to do it. So I'm hoping that maybe outside of this meeting, somebody from our advisory board might step up and be willing to give about a half-an-hour talk